Hi, everyone. Welcome to our video on protein structure. So our goals in this video are really to understand and recognize the different levels of protein structure and to start thinking about the forces that are responsible for those structures. So the first thing we'll talk about is peptide bond formation. Very briefly, at the end of the last video, we looked at a polypeptide sequence and we identified those peptide bonds, or they could be called amide bonds. Okay, and this is a condensation reaction. So as two amino acids come together, so we've got one amino acid, a second amino acid, the carboxyl end of one reacts with the amino end of another, and we're going to see a water get spit off. So it'll grab one of the oxygens from the carboxyl group and a couple of the hydrogens from the amino group. And then we are left with this new bond over here between the two. If we think about the amino acid sequence, as we write amino acid sequence, we're always going to write them from the amino terminal end to the carboxyl terminal end. And the order of the amino acids is the primary sequence. We'll talk about that in just a minute, but that order of amino acids. So when I look at this picture and I look at this and I say, wow, that's tyrosine. All right, that's my first amino acid. It's at my amino terminal. Oh, I can keep going, right? There's glycine. That's my next one. And next one. Up, phenylalanine and leucine. And I can identify my amino acid sequence here. Let's see. If we look at this, we can see each of our peptide bonds. Okay, so here's a peptide bond. There's a peptide bond. There's a peptide bond. There's a peptide bond. When we refer to amino acids that are already bound in the sequence, we call them residues. So amino acid residues, it's kind of the residue that's left after the water splits off. All right, let's take a look at those peptide bonds. And the key thing I want to point out is that those things are planar, that there is not free rotation. So here's that peptide bond. And there's not free rotation, and that's because of this resonance down here, right? That it has partial double bond character. So no rotation around that, but there is possible rotation around other bonds in there. Okay, so if we look at our peptide bond, let me highlight our peptide bond again. There's one of our peptide bonds. Here's one of our peptide bonds. We would not expect to have rotation around those, but you can have rotation around the alpha carbon to nitrogen bond. Okay, we're going to call that the phi bond, or the alpha carbon to the carbonyl bond. Okay, the psi bond. I don't care if you know phi and psi, but it's just important to know that this molecule does have some rotation. The amount of free rotation around those bonds will really be dependent on the amino acid side chain. What is this side chain that is on each of these? That will really help. That steric hindrance will limit rotation and help determine the structure of our molecule. Okay, so primary structure. I thought I had uh, another slide, but I don't think I do. So the primary structure is just the amino acid sequence. What order are those amino acids bonded together in? Once we have that, we can start thinking about the secondary structure. So the secondary structure is the three-dimensional structure formed by the hydrogen bonds between the backbone. So if we think about polypeptide chain. Let's go. Oh, here. I've got one down here. All right. Let's just think about a polypeptide chain. Don't worry about the picture over on the left yet. So we have a chain. We've got our peptide bonds. Okay, the boxes wouldn't normally be there, but just to show you each of our individual amino acids. Each of those amino acid residues has both the NH group and the carbonyl group, right? And each of those has some R group. And they're just by just saying the numbers, they're just trying to say that these don't necessarily all have to be the same. As we talk about the primary structure is just the order of these amino acids. The secondary structure is how this backbone, so we're going to have a repeating backbone that's essentially nitrogen, alpha carbon, carbonyl. Nitrogen, alpha carbon, carbonyl carbon. Nitrogen, alpha carbon, carbonyl carbon, so on and so forth. So this backbone piece, this is what we're looking at when we talk about secondary structure. How is that part that I've highlighted in pink kind of twisted or wrapped around? We're not going to be thinking about the R groups. The secondary structure will be stabilized generally by hydrogen bonding from the backbone, part I highlighted in pink, the backbone carbonyl groups and NH groups. Okay, we're not looking at any interactions between those R groups yet. So one option, one very common thing is what we call the alpha helix, this right-handed helix. And when we look at that, we get our backbone, that this would be essentially be the backbone. Whoops. What do I want here? All right. So here's our backbone, right? Going up there. And then among about every fourth amino acid, we're going to get hydrogen bonding. Okay, between, and we've highlighted some of those here, between this carbonyl and this hydrogen, because that twisting puts them in position to interact. So we get a bunch of hydrogen bonding that keeps us in the coil. Right? We've got some pictures of that followed up here. When we look at this, we've got our alpha helix, right? So the backbone's kind of going, whoops, I don't want to draw it that way though. It's really twisting around, just hard to draw. So that was that pink part we highlighted in the backbone. When you look, you can see that those side groups, the R groups, are in green. And you can see that they're all out to the side because there's not a lot of room down the middle. Okay, you can see the same thing here. This is looking down as if we were looking down the middle of this. And you can see we have this small open area with all those R groups on the outside. Okay, the pink is your oxygen, the gray is your carbon, the blue is your nitrogen, the white is your hydrogen. All right, so what we see, here's a hydrogen bond, hydrogen to oxygen, oxygen to hydrogen. Okay, we see those over and over. So here's our ball and stick model, and here's kind of our space filling model. So you can see that it's really kind of crowded in here. So alpha helix, very common secondary structure, very common way that the backbone lines up. Another is a beta sheet, okay? And you can have a couple types of beta sheets. You can have a parallel beta sheet, parallel, I need that last A there, okay, where we have our N terminal to our C terminal. Notice N terminal to C terminal. These two sheets right here are going the same way. And we see we get those hydrogen bonds set up like that. Now, the other option is a anti-parallel where they're going backwards. And you see that with these bottom two. We have one strip going to the right, nitrogen to carbon, but now this one, whoops, is going nitrogen to carbon this way. And there'd have to be some way that it would wrap around, right? So that it could end up going back the other way. And we get, again, though, key is that we still have hydrogen bonds. And as we indicated up here, we could have a mixed sheet that has some of our beta strands, some of our strands going parallel, some of them going anti-parallel. All right. So one thing to notice, when we're up here in the alpha helix, the hydrogen bonding is near between two amino acids fairly close to each other in the chain. But in the beta sheet, those amino acids, the hydrogen bonding that's holding them in this beta pleated sheet, can be widely varying. They can be very far apart in the chain. Beta sheet could have two chains up to like 10 maybe, forming this big sheet. Okay, a couple just examples to look at. On the left, a fatty acid binding protein. When you see this kind of arrow, we're indicating this strand that's part of a beta pleated sheet, or we just called it a beta sheet in your book here, this beta sheet. And then we'll have these kind of places where it has to twist and turn so that it can form the next beta sheet and get lined up. Ferritin is much an example of one that is mostly alpha helicy. And so you'll see that these alpha helix shown this way in the ribbon. Okay, this has a little bit of that, right? So we can have a mix. Important in these will be what we call reverse turns. And the idea here is that a lot of these proteins are compact and they have to be able to turn back on themselves, right? So here we have a anti-parallel beta sheet, lots of strands, and these things have to be able to kind of turn tightly to get the next piece going. And those turns are often stabilized again with those hydrogen bonds. All right, we are going to have three broad categories of proteins: fibrous, globular, and membrane. And really, for now, we're going to look at fibrous and globular. And we've got a couple examples of fibrous proteins: alpha keratin, okay, which is a structural protein found in wool, hair, is composed of two right-handed alpha helices. So we've got, here's one alpha helix, here's another, and they're essentially in this, what we call coiled coil. They're coiled together. So there's other proteins that have similar structure, but essentially we have these long alpha helices, these long secondary structures that are then twisted together to form even a stronger structure. Okay, another example is collagen. When you look at collagen, it's interesting because collagen has three helices, but they're not alpha helices. They're not that same shape, and they don't have like four amino acids per turn. They're different. And they're a tighter turn, and you need glycine at every third residue to make that happen. Proline is a common piece of this because this is going to be this, it's going to have these tight turns. So you'll see these glycine, proline, oh, maybe something else, glycine, proline. So we have lots of glycine and proline, these small ones that let us do tight turns. Okay, and then collagen has three of them entwined together. That would be this kind of extended secondary structure. And we're still calling it secondary structure because the interactions are all still among the backbone. We're not talking about those R groups yet. I like talking about the collagen because let's talk about a couple interesting facts here. So we said you really need glycine for that. So osteogenesis imperfecta or brittle bone disease occurs if you have a mutation that results in substitution of another amino. All right, so this again, this osteogenesis imperfecta occurs when you have a mutation that has something besides glycine in a spot where there should be glycine. And what that does is it disrupts this structure, these tight turns. You can kind of see on this blue one, you've got these tight turns. And they can't do that if there's something bigger than glycine. Remember, glycine's our smallest amino acid. Another um, collagen interesting fact. So hydroxyproline is a modified version of proline in which you have a hydroxyl group that replaces a hydrogen. And it turns out that this hydroxyproline is actually what's used in a lot of this structure. And it's important to stabilize it because it allows for more hydrogen bonding. So you've heard of scurvy. Vitamin C is required for the formation of hydroxyproline. So lack of vitamin C means we can't form hydroxyproline and we get scurvy. So if you come back up here, collagen, skin, teeth, bones, cartilage. So it's really important. And hence we see issues with bones and with many of those when our primary sequence is disrupted in some way that then disrupts the secondary sequence. All right, onward, tertiary structure. So tertiary structure, now for fibrous proteins, we really just talk about primary and secondary structure. But once we get to globular proteins, we're going to need to talk about tertiary structure, which is how the protein folds up on itself. A great example is myoglobin, which we looked at earlier. So we've got our ribbon drawing and we've got our space filling model. We see we have all these alpha helices, but they've got to be able to wrap around. They fold around. There's very little empty space in a globular protein. These things wrap around on themselves. That's the tertiary structure. So the secondary structure, this alpha helice, right, is held together by hydrogen bonding across the backbone from one backbone atom to another. The tertiary structure is held together by interactions among those R groups. So we're going to talk about ion-ion, we're going to talk about hydrogen bonding, dispersion forces. Those R group interactions define our tertiary structure, how this thing folds up. So as we talk about that, we have tertiary structure. Most globular proteins have complicated tertiary structures. They are very compact. We saw that with our space filling model, little to no empty space in the interior. And generally, because our bodies are largely water, the interior of the protein will have the hydrophobic amino acids kind of hiding in there away from the aqueous areas. And the exterior will have the charged and polar amino acids. So as it folds, it's hiding away and grouping those hydrophobic amino acids more towards the interior. Okay, a couple things, just other vocabulary we want to talk about. A motif or super secondary structure are kind of combinations of secondary structures that are found in many proteins. So, for example, within this larger protein, we see this helix section and then this turn and then the helix section. So, this helix turn helix, that would be a motif that we would see repeated in a lot of different proteins. Another thing is that a single uh, protein, so one long polypeptide chain that goes from N-terminal to C-terminal, sometimes we'll have a bunch of sections that look similar. So even though this is one polypeptide chain, we can see, wow, I've got this looks a lot like that, looks a lot like that, looks a lot like that. Those are called domains. And oftentimes each of those domains will perform a function and they'll all be similar because they have similar structures. All right, we've done primary, secondary, tertiary. Now we get to quaternary. So as we look at quaternary structure, now this is only utilized for proteins that need more than one polypeptide chain. So each of those individual polypeptide chains would be called a subunit. So you would have multiple sheets, each with its own N terminal and its own C terminal. You could have two, you could have three, you could have however many. And so each one of them has their own individual secondary structure, their own tertiary structure, but now they're going to come together in a specific way for the quaternary structure. An example, or here, one example. So these things will be held together by some sort of IMFs, and they're really only functional. This protein will only be functional when you have, in this case, both of these strands. Okay, an example is human hemoglobin. We call it an tetramer because there's four strands. Two are shown in yellow, two are shown in pink. So the yellow ones, there's two identical strands. The pink ones, there's two identical strands. And hemoglobin, which we'll look at a lot more later, is only functional when you have all four strands. Each has, by the way, we can look at it, a heme glute there, there, and there to help carry oxygen. All right, couple more things. Bovine ribonuclease. So ribonuclease is a type of enzyme that cleaves individual nucleotides in RNA. We'll be looking at some of these, but we know that there is a relationship between the primary structure, the order of amino acids, and the tertiary structure, how they all fold in on themselves. But nobody had proven that until this classic experiment from the 1950s. And here's what they did essentially. So when you look at this, and by the way, this would be a great practice to see if you know all your one-letter abbreviations. Go through and see if you can name all of these. So this protein, when you look at it, has all these amino acids and it's folded in on itself. And notice that we have cysteines periodically. And cysteine is interesting because what happens is so we make our strand and then post-translationally after that strand is made, these cysteines when they come into contact can interact and make a disulfide bond, and you see that over here. So you've got the cysteine, you've got the other cysteine, and they're coming together to form that disulfide bond, and that disulfide bond can serve to stabilize the structure. Okay, notice that we're giving off electrons. This is an oxidation process, loss of electrons. So here was the experiment back in the 1950s. They isolated this, put it in a test tube, and then they denatured it, meaning that they interrupted all the intermolecular forces so it would unfold. They did that by adding urea in high concentrations. So that breaks apart all the IMFs, hydrogen bonding, all of that. They also added the beta mercaptoethanol, which was a reducing agent. So it broke the disulfide bond. So now there's nothing holding this in its tertiary structure. And you get this kind of just random, right? Uh, Denatured amino acid. So the amino acid chain's still intact, but it's not in any specific order, or not in any specific three-dimensional arrangement. The order of the amino acids doesn't change. So when it's denatured, they found, hey, no surprise, the protein no longer serves its function. It's not working as this enzyme when it's denatured. So the experiment was like, hey, if we take this back to its physiological conditions, If we get rid of the urea, we get rid of the beta mercaptoethanol, we make sure it's in the appropriate pH, will the protein become functional again? And it turned out, yeah, yes, that primary sequence is what determines that tertiary sequence. So once we got rid of all the things that had disrupted it, it naturally folded back into its exact perfect protein, three-dimensional structure again, and was functional. So it proved that just based on the amino acid sequence in vitro, in test tube, we can denature and renature it, that amino acid sequence is strongly aligned with the function. And this also tells us small changes in the amino acid sequence can disrupt function, and we'll be looking at some of that. All right, last thing for this video is just a couple quick notes, so I don't leave you with the wrong impression that everything is perfectly ordered and all the same for every single protein. Here we go. There are some intrinsically disordered proteins that essentially don't have a defined structure until they interact with other molecules. There are also metamorphic proteins that exist in a couple different structures. And the structures are fairly different there, right? And depending on the molecules they're interacting with, depending on the environment, one or the other will be the primary form. All right, that is it for protein structure. And next, we're going to look at kind of how we can isolate and separate proteins.